Hello, good evening, everyone. Welcome to lecture one on cell cycle and cell division. So, in this lecture today, we will be covering about what are the characteristics of a cell growth, what are the basic steps in cell cycle and division, and also we will have a very short discussion about the type of division, which is mitosis and meiosis. So this will be the today's topic that we will be discussing in today's lecture. What is the basic characteristic of living organism? We know that a living organism has two very important characteristics. Firstly, it grows, and secondly, it reproduces. Right? Mule, which does not reproduce, but the basic characteristic of cells, or we can say of all the living organisms, is that it undergoes growth and it undergoes reproduction. The growth should be internal as well as external, and reproduction should take place. For example, if you can say that there is some dust which is deposited on the surface, so you might say that with time it may become a heap of the dust. This is not the growth. This growth is external to the accumulation growth. The growth should be internal, and organism should reproduce. So these are the two basic characteristics of any. A cell has to divide. Right? So a cell reproduces by dividing it into the two. So for example, there is a single cell. What we can say that it reproduces or it grows by dividing it into the two. So this is a parental cell that contains some of its DNA, and then these are the daughter cells with the DNA. So these are the daughter cells, right? Again, these daughter cells, upon growth, they will grow with time, and then again they will further reproduce to give rise to two other daughter cells. So this is how the cells reproduce by dividing it into the two, with each parental cell giving rise to the two daughters. Cells happen by the process which is known as the cell division. Cell division is basically the growth and the reproduction in the cells. So these newly formed daughter cells can further, as I mentioned, can grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. So as I showed you in the previous diagram also, where we had one parent cell. So for parent cell, I'm using the notation PC. The parent cell give rise to the two daughter cells, right? And what this statement means is that the daughter cells further grow. So they grow in size. So let's say they grow in size. They come to a size when they can further reproduce. So these daughter cells will now again undergo the reproduction, giving rise to the two new daughter cells. Now, in this case, for these daughter cells, these will be the parental cells, right? So this is the case that the newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell. So this is formed by the growth and division of a single parental cell and its progeny. So these are their progeny, right? These are the progeny of these grow and reproduce. So this will continue. So this is how the reproduction in the cells happen, that the parental cell give rise to daughter cells, which can be called as progeny, which further undergoes growth and give rise to further new daughter cells, and the continues. So this is how the reproduction in this and division allow a single cell to form a structure consisting of millions of cells. So this is how the cell division happens. And a single cell, we all know that our life starts from a single cell, which is the zygote. After the formation of zygote, many different cells are formed by the process of cell growth and cell division. A cell reproduces by performing an orderly sequence of events in which it duplicates its content. Since it has to reproduce, so we know that a cell has some DNA also and other components as well. So before division, it has to duplicate all its content so that it can provide the same content to the daughter cells. So it has to duplicate the content. So it has to duplicate its contents, and then it divides into the two. Right? So firstly, it has to undergo duplication and then it has to divide. So this cell cycle or this cycle of duplication and division is known as the cell cycle. And this cell cycle is the essential mechanism by which all living things reproduce. So the division in the cells happen with the help of the process which is known as the cell cycle that involves the duplication of the content and then the division. Fine. So this is the cell cycle. So what is the basic introduction? That in these cells to grow, growth and reproduction are the two basic characteristics. And a single cell can give rise to multiple cells by the process of cell division, in which firstly the cells grow, accumulate its contents, and come up to a size where it can undergo reproduction or cell division. And this cycle of duplication and division to give rise to the new daughter cells is known as the cell cycle. And it is an essential mechanism by which all the living things reproduce. So this is the basic introduction. Then we have two different types of species. Firstly, they are the unicellular and the second one are the multicellular. So what are unicellular species? As the name suggests, uni means single. So these are single celled species. Right? And then multicellular means having many cells. So in unicellular species, for example, we know bacteria and yeast, each cell division produces a completely new organism since they are only made up of cells. So what we can say is that suppose this is the bacteria, which is a unicellular organism. So the bacteria can give rise to the first. So each cell division, so basically a bacteria give rise to the daughter cells. So the bacteria give rise to the daughter cells, and these daughter cells are in turn nothing but the new bacterial cells. Fine. So for the unicellular species, so this is the parental cell. This parental cell can give rise to the daughter cells. These daughter cells are indeed a new organism. Right? So the cell cycle produces a new organism. This is how the reproduction happens in the unicellular organisms. So in unicellular species such as each cell division produces a completely required to form a functioning organism. A single cell division is not enough to produce the complete organism, to produce functioning organism, right? So in this, a series of cell division is required to produce a functioning organism in multicellular species. is formed, cell division stops. So even in the adult body, once the functioning organism is formed, the growth happens by the process of cell division. So cell division is usually needed to replace the cells that die, because we know that cells have a limited time period for which they can survive. After which, once they die, they have to be replaced by the new cells. So, even in the adult body, cell division is usually needed to replace the cells that die. And cell division is of two types, as I mentioned in the introduction, mitosis and meiosis. So, we will be studying about mitosis and meiosis one by one. Now, but what is the basic characteristic of mitosis and meiosis? So, these are very important points to remember, that mitosis produces genetically identical cells. So the cells which are produced are genetically identical. Their genetic material is same as the parental cell, right? While in meiosis results in variation. How this happens? So by a process of crossing over. So this we will see in the later lectures, but meiosis results in variation and gametes are formed of meiosis are formed. So the formation of gametes takes place with the help of meiosis. Sperm and ova, for example, I considered humans, but it may be other in other species. So sperm and ova, which are the gametes, are formed by the process of meiosis. And meiosis results in variation, while in mitosis, the cells are genetically identical to their mother cells. Their genetic material or the DNA or the genetic content is same as their mother cell or the parental cell. So these are the very important points to remember in mitosis and meiosis, that it does not result in no variation. In this, the variation happens. So giving rise to the diversity. So what is cell cycle? So we know that cell division is a very important process in all living organisms. And during the division of a cell, DNA replication. So what are the processes that takes place during the division? Firstly, DNA has to replicate. So DNA replication and cell growth also takes place. So all these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. So what are the three processes that happens? And these three have to be in a very coordinated manner. So they are DNA replication. So maybe we can start with the cell growth. Firstly, a cell has to grow. So cell growth, DNA replication, and then cell division. So all these has to take place in a very coordinated manner, and this complete process is known as a cell cycle. So the stage through which a cell passes from one division to the next is called the cell cycle. As I mentioned, it is a sequence of events by which a cell duplicates its genome, synthesize the other constituents, like for example some proteins which are required or the lipid which is required, and eventually divides into the two daughter cells, and this complete process is known as the cell cycle. So in which it involves the duplication of the genome, which it can pass it to its progeny, then the synthesis of other components and the complete cell growth, and finally the cell division, dividing it into eventually into two daughter cells. Although cell growth in terms of cytoplasmic increase usually is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle that we will see. The increase in the amount of cytoplasm is a continuous process. It's a continuous process during the cell growth. But when does DNA synthesis or DNA duplication has to take place or DNA replication has to take place is very specific. And the error in any of these stages may lead to diseases like cancer. etc. So therefore, it has to be in a very coordinated manner, and it takes place in a very specific manner. So the replicated chromosomes DNA are then distributed to the daughter cells by a complex series of events that happens during the cell division. And these events are themselves under the genetic control. They are very controlled events that takes place that allows the division of the cell, passing the genetic material from one generation to the next generation. So this is a representation of a cell cycle. So these so this is the first this is a hypothetical diagram in which a eukaryotic cell with two chromosomes has been shown here. So firstly, what happens is cell growth and chromosome replication takes place. So cell grows in size. You can see it grows in size. DNA duplication happens. You can see that here only the two chromosomes were there. N is equal to 2. Here N is equal to 4. Then chromosome segregation has to happen. So the chromosome will segregate. Still it is N is equal to 4. And once the cell divides, then it again becomes N is equal to. So by the process, these cells divide. This increased amount of volume gets divided into two daughter cells and resulting in the daughter cells which are similar to their parental cells. So the division of a hypothetical eukaryotic cell with two chromosomes has been shown here. And you can see that in the end, the two identical daughter cells are produced, which have the similar genetic material. Now, what are the phases of cell cycle? As I mentioned, different process has to take place. Cell has to grow in size. Cell has to duplicate its DNA content. Cell has to accumulate or synthesize other components like proteins, DNA, lipid, etc. So, in what phase, what happens? We have to see that. And finally, the cell has to divide into two cells. So, basically, what is the basic mechanism and what are the different phases? So, a typical eukaryotic cell has been illustrated. And these cells divide once if I talk about the cell culture that we performed in the laboratories. So it approximately take every 24 hours to divide a cell. However, this duration can vary from organism to organism and also from cell type to cell type. For example, there are certain cells that grow very fast and they may become confluent in within 24 hours, while there are other cells that require larger time and you have to incubate them maybe for 48 hours. So yeast, for example, if I like we know that for E. coli, the division time is 18 minutes. While if I talk about the yeast, it can progress through the cell cycle in only about 90 minutes. While if I talk about the eukaryotic, it is usually like eukaryotic human cells or some other organism. Usually it takes every 24 hours. So there are various stages as you can see here. So first one is the G1 stage. Then we have quiescent stage also, G0, which is the quiescent stage. Then we have S phase, which is the synthesis phase. We have G2 phase, second growth phase. Then in the M phase, different. So this M phase helps in the other processes where the synthesis of other components and the division of the components into two daughter cells happen. So what are the phases in the M phase? So it is further divided into four different phases: prophase, metaphase, anaphase, and telophase. Finally, the cells divide by the process of cytokinesis. Kinesis means division, cyto means cell. So cell divided into two parts, two daughter cells, which is cytokinesis. So starting from the one single cell, we have two daughter cells. Also, in order to remember the different phases of M phase, you can remember pre-medical admission test. So this is a mnemonic that will help you in remembering the order of the phases that happens in the M phase. So first is the pre, that is the prophase. Medical, M stands for metaphase. Then admission, that stands for anaphase. And test, that stands for telophase. Right? So these are the different phases of the within the M phase where different processes happen, and that we will see what are these different steps. So the cell divide. So basically, the cell cycle is divided into two phases. One is from this G1, S, and G2 phase are together called as the. So from here to here, just a second now. So if I have to talk about the different phases, then the cell cycle consists of two phases. From here, that is G1, S, and G2. It is called as the interphase. And the second is the M phase, which is the mitosis phase in which the division happens. Right? So this is the interphase and the M phase. So this is a diagram which is shown the duration in hours of the four stages vary. This is just a hypothetical diagram showing if a cell has to spend 24 hours, how much time approximately it will spend in the M phase, how much it will spend in the G1 phase, how much in the S phase, and how much in the G2 phase. So just to introduce you from the G1, from this side, from this point to this point, it is the interphase. And then this is the M phase, right? So first one is the G1 phase in which a cell spends almost 6 to 12 hours if it is a 24-hour cell cycle, right? So G1 phase, in what happens in G1 phase? It is a growth phase. So it is a growth phase. So basically RNA and protein synthesis happens, no DNA replication or no DNA synthesis happens, right? So there is no DNA synthesis, only RNA and protein synthesis happen. You need to remember what is actually happening in what phase. So in G1 phase, RNA and protein synthesis, right? In this, we have a G0 point also, which is the quiescent stage. It is also known as the G0 phase is the terminally differentiated cells withdraw from the cell cycles indefinitely, and they do not enter into the cell cycle. So sometimes what happens is that the cells which have become completely differentiated, which have acquired the function, now if they do not have to divide further, for example, neuronal cells, we know that the neuronal cells do not divide or they have a very limited division capacity. So they enters into the G0 phase, which is the G0 phase in which the cells do not divide. And although there is a re-entry point, a cell returning from G0 enters at the early G1 phase, right? Then there is a restriction point only after a certain time point when there is enough RNA and protein synthesis. A cell that passes this restriction point is committed to pass into the S phase. This S phase is known as the synthesis phase, in which the DNA synthesis happens. So that is why it is called as the synthesis phase, in which the DNA synthesis happens. Doubles the amount of DNA in the cell. RNA and protein are also synthesized in this phase. Then comes the G2 phase, which is the second growth phase. So in this, no DNA synthesis happen, but RNA and protein synthesis continue. The growth of the cells continue, but the replication or the division of duplication of the DNA happens in the S phase. So DNA replication happens in the S phase, which is the synthesis phase. Then comes the M phase, which is the mitosis, nuclear division, and cytokinesis, which is the cell division, yields into the two daughter cells. What are the steps of the M phase? As I mentioned, there are 4 steps. The first one is prophase, metaphase, anaphase, and telophase. Right? So pre-medical admission test, that is prophase, metaphase, anaphase, and telophase, followed by cytokinesis that leads to the division of the cell. So starting with the interphase that involves G1, S, and G2 phase. So period of preparation for cell, it is a period of preparation for the cell division, and we can say that it is the phase between two successive M phase. So from one M phase to the another M phase, it is the, so suppose this is the M phase. So suppose this is the M phase, in which, so the period of cycle from this to this, from one M phase to starting point of one M phase to the end point of the other M phase, between the two successive M phase, is known as the interphase. What is happening in the interphase? The cell is growing in size, it is preparing itself for the next division, and it is actually the most active phase of the cell cycle, in which the cell is actively working. It is significant to note that if we are considering 24 hour of average duration of the human cell, more than 95% of the duration is spent in the uh, interphase, and only the cell division only lasts for only about an hour. However, you can see that M phase is only for about an hour, while the remaining time, which is the 95% of the time, cell grow, invest into the cell growth and DNA replication, etc. So 95% of the duration of the cell cycle is the interphase. During this phase, a series of metabolic changes occurs, and these changes are not visible under the microscope. So therefore, some scientists termed interphase as resting phase, but in actual, it is the most active phase. But in actual, it is the most active phase. It is the time during which the cell is preparing for division by undergoing both cell growth and, as I mentioned, DNA replication in an orderly manner. There are three different phases, G1, S, and G2, that happens in the interphase. So, as I mentioned, the interphase is further divided into three further phases. First is the gap 1, S phase synthesis, and the second is the gap 2. So, partly to allow time for growth, most cell cycle have gap phases, or in which the growth happens. A gap 1 phase between M phase and S phase, and G2 phase between the S phase and meiosis. So we know mitosis, sorry. So we know that if I have to start a cell cycle, it is G1, S, G2, and M. So G1 is between the M and S phase, and G2 is between the S and M phase. Right? So thus the eukaryotic cell is traditionally divided into four sequential phases, G1, S, G2, and M. G1, S, and G2 are together called as the interphase. So as I mentioned, this complete process from here to here is called as the interphase, and the remaining one is the M phase, where the cell division happens. In a typical human cell, which we are growing in the cell culture, as I mentioned, that it interface 95% of the time is dedicated to the interphase, right? So in a typical human cell, which we are growing in the cell culture, 95% of the time is spent in the interphase. So starting with what is happening in the G1 phase, which is the gap 1. So G1 phase corresponds to the interval between the mitosis and initiation of DNA replication. We know that initiation of DNA replication happens in the S phase, and mitosis is the M phase. So basically, G1 is the time point between M and S phase. So during G1 phase, cell is metabolically active. It continuously grows, but it does not replicate its DNA. This is very important. G1 phase is the period when the cell grows and carries out normal metabolism. Most of the organelle duplication also occurs during this phase. So most of the duplication, like mitochondria, for example, it has to duplicate. Other organelles have to duplicate. So most of the organelle duplication also occurs during this phase. And during G1, cell rapidly synthesize different types of RNA and proteins that I have already mentioned. And due to the availability of proteins, synthesis of new protoplasm takes place. Size growth happens during this cycle. Because of the availability of the protein, the protoplasm increases and size growth happens. So cell grows maximum in the G1 stage. This is very important, that maximum growth happens in the G1 stage. Next phase is the S phase, which is also known as the synthesis phase. As I mentioned, it is a DNA synthesis phase in which the DNA replication happens, right? So replication of nuclear DNA and synthesis of histone proteins. Histone proteins are the ones that binds with the DNA and helps in making it compact. Right? It takes place in the S phase. Replication of cytoplasmic DNA may occur in any stage of the cell cycle. This is very important, that cytoplasmic DNA may duplicate in any phase of the cell cycle, while the replication of nuclear DNA takes place only in the S phase. During this time, the amount of this statement is really important. You have to focus on this as questions are usually asked. During this time, the amount of DNA per cell doubles. So suppose if the initial amount of DNA is denoted as 2C, then the total amount of DNA after the S phase will become 4C, because we know in this phase DNA replication is happening. So whatever, suppose we have this 2C content, after this we will have the 4C content. This is the after S phase. Before S phase, it will be just 2C content. So after S phase, since the DNA replication has happened, so if I say that the total amount of DNA in the before the S phase is 2C, then after the S phase it will be 4C. However, there is no increase in the chromosome number. This you have to remember, that the chromosome number does not increase. If the cell had diploid or 2n number of chromosomes at G1, even at the S phase, the number of chromosomes remains the same. So this is important, that the chromosome number remain the same, while the DNA content doubles. This is important, that the chromosome number remains the same. This is actually, if I have to make a true representation, so I have to make it like this, that the DNA content is 2C, but the chromosome content is, the DNA content becomes 4C, but the chromosome content is still the same, because they remain attached to each other. During the S phase, it does not divide. So therefore, the chromosome number, this is very important line. This is usually asked in the exams also, that the chromosome number, if the chromosome number is 2n, then even after the S phase, the chromosome number will be 2n only, but the amount of DNA per cell doubles from 2C to the 4C. S phase marks the synthesis of DNA replication and chromosome duplication. DNA content in a chromosome becomes double. So basically, within a chromosome, the DNA content doubles. So within a chromosome, the DNA content doubles. Therefore, number of chromosome, as I already mentioned, number of chromosome remains same, but DNA content doubles. In animal cells, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. So this is important, because in animal cells, we have a special structure which is the centriole. So this centriole duplicates in the cytoplasm during the S phase, while the DNA replication begins in the nucleus. The next phase is the gap 2 phase, which is the G2 phase, in which also the growth happens. So it is a period of cytoplasmic growth. Final preparation occurs during this phase. Special materials which are, for example, tubulin protein, which is required for the formation of spindle fibers in the M phase, are synthesized in the G2 phase. And cell growth continues during this phase. So basically, this is the G2 phase. Now, as I mentioned, there are certain cells which entered into the G0 phase. So some cells in the adult animals do not appear to exhibit division. So they stop cell division, and many other cells divide occasionally. So some permanently remain in G0 phase, while some re-enters into the from G0 to G1 phase. So these cells do not further exit G1 phase to enter, and they enter an inactive stage, which is called as the quiescent stage or G0 stage of the cell cycle. Cell in this stage remains metabolically active as the other cells, but it no longer divides, or it no longer proliferate unless called on, depending upon the requirements. If they are called on, then they re-enters into the G1 phase. Otherwise, they remain in the quiescent stage, in which the cell is metabolically active, but it is not undergoing any sort of division. Then the last phase, which is the division phase or the M phase. So in this, or it is also called as the mitotic phase, which lasts only about an hour in the 24-hour cell cycle. It represents a phase when the actual cell division or mitosis occurs. During this phase, cells actually divide into the two cells. In animal cells, mitotic cell division is restricted or only seen in diploid somatic cells, except in some social insects. Against this, the plants can show mitotic division in both haploid and diploid cells. Otherwise, in animal cells, in diploid cells, usually what happens is, usually it is meiosis, but in diploid somatic cells, it is the mitotic cell division. This is the most dramatic period, involving a major reorganization of all the components. And since the number of chromosomes in the parent and progeny cells is the same, it is also called as the equatorial division. So mitosis is also known as equatorial division, right? Because the number of chromosomes, when we will see in parents and progeny are the same, right? So the M phase starts with the nuclear division, corresponding to the separation of the daughter chromosome, which is known as the karyokinesis, and usually ends with the division of cytoplasm, which is the cytokinesis. So karyo word is related to nucleus. So firstly, it involves the division of nucleus, then the division of cytoplasm. So mitosis, as I mentioned, has been divided into 4 stages of nuclear division: prophase, metaphase, anaphase, and telophase. So these are the stages of karyokinesis, which is followed by cytokinesis. Right? So starting with the prophase. So prophase is the first phase of the M phase. In this, you can see that what happens in the prophase is, at prophase, the replicated chromosome, each consisting of 2 closely associated sister chromatids, condense. So as I mentioned, DNA replication happens, but the chromosome remains the same. So what will be the earlier stage of this cell? So earlier, this cell will contain only these 3 structures. But during the S phase, what would have happened? That duplication has happened, but these sister chromatids which are formed, which are similar to the previous DNA, remains attached. So these are known as the sister chromatids. So this is a sister chromatid. In this, the nuclear envelope is intact. Kinetochore is the structure which helps in keeping both the chromatids together. And then we have centrosome forming the mitotic. These are the forming mitotic spindles. So outside the nucleus, the mitotic spindle assembles. So you can see that outside the nucleus, the mitotic spindle assembly is taking place between the 2 centrosomes. So we have 2 centrosomes between which the mitotic spindle assembly is taking place, and these have replicated and moved apart. In diploid cells, there would be 2 copies of each chromosome present, as I mentioned here. You can see in this figure. Then the next phase is a metaphase. So what happens in metaphase is, you can see that centrosomes at the spindle poles. So basically, the centrosomes reaches to the pole. They are pole apart, and these structures are known as the kinetochore microtubules that attaches to the kinetochore, and you can see that these are the 2 centrosomes. This is also a centrosome, which is a duplicated centrosome. So these centrosomes moved apart, and you can see that the kinetochore are attached to the opposite sides of the 2 sister chromatids. So the chromosomes are aligned at the equatorial plate and mid is between spindle pros. So the kinesical, while the other side is attached to the other side of the other centrosome. So in the metaphase, what is happening is that the chromosomes are aligned. In the metaphase, the chromosomes are aligned on the centrosomes at the spindle. Centrosomes reach the spindle pole, and the chromosomes are aligned themselves at the axis, central axis. You can see here. And the kinetochore microtubules help in the attachment of these centrosome with the one end of the sister chromatids. So these sister chromatids are attached to the opposite poles, and once a force will be applied, they will move apart from each other. That will happen in the next phase. So basically, at metaphase, the chromosomes are aligned at the equator. So this is known as the equator of the cell. You can say this as the equator of the cell or equator of the spindle, which is midway between the spindle poles. And the kinetochore microtubules attach sister chromatids to the opposite poles of the spindle. So these you can see here that if I zoomed in into this, these kinetochore microtubules, which are shown in the green color, are attached with the red color kinetochore on the opposite side of the sister chromatids. So this happens in the metaphase. Then the next phase is the anaphase. As I mentioned, that once the force is applied, since the kinetochore microtubules are attached to one side of the sister chromatids, the shortening of the kinetochore microtubules applied a force due to which these two sister chromatids get separated from each other, and now they are called as the daughter chromosome. So basically, if the chromosomal content during the metaphases, suppose for example 2C, the chromosomal content now will become the number of chromosomes, not the chromosomal content. Not the chromosomal content, but the number of chromosomes. Suppose here it's 3. Now it will become n is equal to 6. So now the number of chromosomes have increases. This is very important. That during the anaphase, the number of chromosomes doubles. At anaphase, the sister chromatids synchronously separate to form two daughter chromosomes, and each is pulled towards the spindle pole it faces. So one moves towards the one side of its spindle pole, the other moves the opposite. The kinetochore microtubules get shorter and the spindle poles also move apart. Both processes contribute to the chromosomal segregation. So basically in anaphase, what is happening? In anaphase, chromosomal segregation happens. Right? So chromosomes segregate from each other. And in what happens in telophase? In telophase, the two sets of daughter chromosomes arrive at the spindle pole. You can see here that the nuclear envelopes start assembling and the interpolar microtubules are like the two daughter chromosomes reaches to the spindle pole and the nuclear membrane starts reassembling. The contractile ring starting to contract. So this contractile ring starts to contract, resulting further in the cytokinesis. And a new nuclear envelope reassembles around each set, completing the formation of two nuclei. So basically, here what has happened? Telophase karyokinesis has happened. Because a single nucleus is now divided into two nucleus. One is here and the other is on the other spindle pole. The division of the cytoplasm begins with the contraction of the contractile ring. So this is just the starting of the contraction of the contractile ring. After this, the division of the cytoplasm happens in the process which is known as the cytokinesis. So cytokinesis is nothing but it is the division of the cytoplasm. So complete nuclear envelope surrounds the decondensing chromosome. You can see here the complete two nuclei are now formed. Contractile ring create cleavage furrow, and due to which you can see that the cell finally ultimately divided into the two daughter cells. So during cytokinesis, the cytoplasm is divided into by a contractile ring of actin and myosin filaments. So this is a contractile ring which is formed by the actin and myosin filament, which ultimately pinches off. So ultimately it will pinch off the cell into the creating two daughter cells, each with one nucleus. So in both the two daughter cells, we will have one nucleus. So this is the basic steps of the cell cycle, that how and when the cell division happens, how and when the nucleus divide, how and when the genetic material divides. So this is the basic steps. Now we will practice some practice questions quickly so that we can have a quick revision of whatever we have understood today. So DNA synthesis takes place during which phase? So we know that DNA synthesis happens during the S phase, which is also known as the DNA synthesis phase of the cell cycle. So therefore, the correct answer is option number 2. The second question is, which of the following is the correct sequence? We know that starting from the G1, it is G1, then S, which is the synthesis phase, then gap 2 phase, and then it is the M phase. So the correct answer will be G1, then S, then G2, and then M, which is the mitotic phase. So the correct answer is option number 4. The third is, which phase of the cell cycle is the quiescent stage? We know that in the quiescent stage, cell does not divide. And this phase is represented by the G0 phase, which is the quiescent phase. So therefore, the correct answer is option number 2. The next question is, during which of the following phase does the assembly of mitotic apparatus begins? So we know that there are 4 phases: prophase, metaphase, anaphase, and telophase. So the assembly of mitotic apparatus begins from the prophase. First phase is the prophase of the M phase. So M phase is divided into karyokinesis followed by cytokinesis, right? That involves prophase, prophase, metaphase, then we have anaphase, and then we have telophase, right? So it starts from the prophase. Sequence of events taking place during M phase. So in M phase, as I mentioned, there are 4 phases: pre-medical admission test. You need to remember this: pre-medical admission test. So prophase, metaphase, anaphase, telophase, and followed by cytokinesis. So prophase, metaphase, anaphase, telophase, and cytokinesis. So the correct answer is option number 3. The next question is, which of the following is the last step of the cell cycle? We know in the last, the cell divides into 2. The cytoplasm divides. So the last step is the cytokinesis. What happens in the synthesis phase during the cell? We know why it is called synthesis phase or S phase, because during this the DNA duplicates. So DNA synthesis happens. We know that the chromosome number does not become double, only the genetic content become double. Therefore, the correct answer is option number 1, DNA synthesis. The next question is, during G2 phase, which is the second gap phase, a diploid cell contains the amount of DNA equal to a diploid cell. So we know that first it starts with the G1, then in S, then in G2. So if in S it is 2C, during S what will happen is DNA content become double. So therefore in the G2 phase the DNA content will be 4C. So if it is a diploid cell that has a 2C content, then in the G2 it has a content equivalent to tetraploid cells, right? So the correct answer will be tetraploid. Sometimes, just to trick students, the examiner may ask that the initial content is 3C. What will be the content in the S phase? So it will be the double of 3C, that will be the 6C, right? So basically that is how the questions might be asked in the exam. The nuclear envelope reappears at which phase? So we know that the phases is prophase, metaphase, anaphase, and then we have a telophase. So the nuclear envelope starts reappearing at what phase? Starts reappearing at anaphase, and then in anaphase the cells, the chromosomal segregation happens, and in telophase the nuclear envelope starts appearing. So the correct answer is option number 4, telophase. Gap between division phase and start of the DNA replication. So start of DNA replication is S phase and division phase is known as the M phase. So the gap between division phase and the S phase is the G1 phase. We know this is the cell cycle. This is the cell cycle. So basically, right? So this is how the cell cycle happens. So this gap is known as the G1 phase. So the correct answer will be the G1 phase. With this, I would like to end today's session. Thank you all for your attention. In the next lecture, we will be understanding about the mitosis, and then we will take up with the meiosis.